This video article demonstrates a procedure for synthesizing a targeting polymeric nanoparticle system for delivery of plasmid to cancer cells. To begin, thiolated gelatin polymers are synthesized and DNA plasmids encoding EGFP are encapsulated. Next, the thiolated nanoparticles are pegylated and EGFR-specific peptides are conjugated to the surface. The nanoparticles are then applied to the cells of interest and incubated to allow protein expression. Analysis by fluorescence microscopy reveals efficient expression of nanoparticle-delivered plasmids as soon as 48 hours after transfection.
The main advantage of this technique over existing methods like delivery of genetic materials using cationic vectors is that gelatin nanoparticles are much less cytotoxic. The implications of this technique extend toward cancer therapy. By targeting on the overexpressed receptors on cancer cells, it's easy to deliver genetic materials into target cells. Though this method can provide insight into plasmid delivery, it can also be used in other systems such as siRNA and microRNA.
Begin this procedure by generating thiolated gelatin. First, dissolve 1 gram of gelatin in 100 milliliters of deionized water and incubate with 20 milligrams of 2-aminothiolene hydrochloride at room temperature for 15 hours. Next, to remove non-reacted reagent, transfer the solution to a dialysis bag and submerge it in 1 liter of 5 millimolar hydrochloric acid solution. After 3 hours, transfer the bag to 1 liter of fresh 5 millimolar